So we go now to osmosis process. So osmosis process is the net movement of water across the selectively permeable membrane, driven by a difference of solute concentrations on two sides of membrane. So it's either the low or higher concent from low to higher concentration of solute. The passage of solvent from less concentrations to a more concentrated solution through a semi-permeable membrane. So it is only the solvent in the form of water that crosses the semi-permeable membrane, because cell membrane are highly permeable to water. So we now to isotonic solutions. When you say isotonic solutions, when exposed to solutions, should keep their normal shape, or it is the osmotic pressure from outside or inside environment of the cells are equal. Okay, or normals. The isotonic solutions, normal. There is a normal size of red blood cells, are round and are biconcave or concave shape. So there is a balance of the movement of water from inside to outside environment of the cell. So the amount of water is transported is equal. Okay, solute concentrations inside of the cell is equal from the outside environment of the cells. So that's the term as isotonic solutions. So normal cells are concave or biconcave in shape. Next, we have hypotonic solution. Hypotonic solutions will have a lower concentration of solute than the cells. So the cells will also have higher osmotic pressures. The tendency of water is to move into the cells because it has a lower concentration of solute, and then the solution surrounding it, this will cause the fluid to move into the cells. So the red blood cells swells, then bursts, and also known as lyse. So the cells inflate, eventually burst. So the water is transported into the cells, and solute concentration inside the cell is higher. Okay. Next, hypotonic solution. The cells shrink. Water is transported outside the cells. So the solution, solute concentration inside the cell is lower. So hyper means too much. Hypertonic solutions will have higher concentration of solutes than the cells, and we have higher osmotic pressures outside the cells than inside the cells. So this will cause water to pull from the cells, which result in attempt to equalize the osmotic pressures. So the tendency of the cell is to shrink. Kasi ang tendency, higher yung concentration inside the cell, and the solute concentration, inside the cell, is lower than outside. So tendency of water is to leave out of the cytoplasm, and the tendency of the red blood cell is to shrink. So these are the difference between hypertonic, isotonic, and hypotonic solutions. Okay, so usually in hypertonic, water leaves the cells due to lower concentration of solute, and we have isotonic, equal, and we have here the higher concentration of solute, so water tends to go inside the cells and tends to the red blood cells burst, and this one, the red blood cells lysis, shrink, and this one, water the red blood cells maintains the concave shape or normal shape of the blood cells. Hydrolysis, the composition of chemical compound reactive with water, such as dissociation of a dissolved salt or catalytic inversion of starch to glucose. So when we say hydrolysis, if you add a water, so it will split the two, the compounds, or split the compound molecules into one molecule plus one molecule. Okay, plus the starch, that is a chain of the glucose plus glucose, that is a starch. So if you add water, it's split, starch will split into glucose plus glucose, like sugar. Okay, table sugar or sucrose is sucrose is composed of fructose plus glucose. If you add water, it will separate into fructose plus glucose. Pancreatin is a mixture of several digestive enzymes produced by exocrine cells of the pancreas. It is composed of amylase, lipase, and protease, which hydrolyze specific biomolecules. Amylase will hydrolyze starch, lipase for lipids, fructose for proteins. Actually, we don't do the experiment about pancreatin because there is no available pancreatin enzymes. Okay. So trypsin, another trypsin is another enzyme found in pancreatin, works to hydrolyze proteins into oligopeptides. When you say oligopeptides, these are proteins that has a less than 25 chain or minimum of 25 chain amino acids. Amylase hydrolyze starch into oligosaccharides, okay, or into glucose plus glucose. Disaccharide, maltose, okay. Maltose is equals to mannose plus mannose, okay. Lipase hydrolyze triglycerides into fatty acids and glycerols. So later on we'll discuss about the pathway of the lipid metabolism process. Okay, pancreatin is an effective enzyme supplement replacing pancreatic enzymes. So aids in digestion of food in cases of pancreatic insufficiency. I mean, if there is a insufficiency in pancreatic juice, so pancreatin is added to to digest food. I mean.